Sorry about that. No, sorry, we are back on track. Okay. Good, perfect. So your OV pop work and then OVCWD, you had to do a few steps in there. And if you can open this web page, the link you have in your email that you got from, yeah. And on the web page, if you go to the web page, I think you can follow these instructions here, right? So you haven't run it for 10 iterations, so you can just follow what we are doing right now. I am just going to ask them to set it on 100 iterations and then come back and actually discuss this stuff. Is that okay? Yeah, sure. Yeah, perfect. Okay. So before, in the last workshop, what we did was we just made sure that OVCWD was running on your computers. You did one iteration and it ran well for all of you. If you can go and check into the results folder, you can see a table written, which I'm going to open up here just to tell you some features of what the OVCWD is writing. Okay, so OVCWD, and please tell me if you cannot see this screen, Zoom. I have already shared it, but I will just make sure that. Okay. Zoom participants can see the Excel, the CSV file open on there. Okay, so the single run that you did, we changed the duration from 25 years to 15 years. And so on the very first column, you have year 1 to year 15 of the model run, of that infection dynamics model, okay? So my point is, I have, again, please keep in mind, I started with this model about 10 years ago. And at that point of time, it was not clear to me what all data I want to document. It's like playing God in this artificial model population. You can document whatever you want, but it will just make a bigger file. So at that point, I selected few of these metrics, and then I also added a few later on. But at any point of time, if we come up with a new question which needs for us to look at something new, it can easily be added to the output file. Okay, so for now, what we have is for every year. So every row represents an year, and we have the post-harvest. We document all the different age classes in the population in the model, right? And so this is the total pre-harvest population. So sorry, the first part is pre-harvest population, just before the hunting season starts. Then you have the adult males infected with CWD, yearling males infected with CWD, and so on. So column I to column N are the 6 sex age classes with number infected in that particular class. Remember, we set up the OVCWD so that a dispersing yearling male walks into our population. So you should see one here, right, in the yearling male infected that walked into our population, and a total CWD infected number of 1. Does that make sense? The very first row, first year, you just had 1 infected there walk into your population. Okay, that is what it is showing. And Zoom participants, please interrupt if there is anything that is not clear. From column P onwards, we have all the harvest data for that year, for each age sex class. So 1, 2, 3, 4, 5, 6, right? Fawn female harvest, yearling female harvest, and adult female harvest, and the 3 classes for male harvest. V onwards, this is something that will get interesting for all of us, because then you have the animals that were tested for CWD. Now, this is based on the, if you go back to the interface of the model, there is a slider. Maybe I will just bring that up. Okay. On the model interface, notice that there is this additional set of sliders. This was not there on our OVPOP model, right? So what do these sliders do? This is percent of the fawn male harvest tested, percent of the fawn female harvest tested. So for the 6 age sex classes, what percent of the harvested animals are tested for CWD? You can set that up. That is mostly available from the agency based on what number were tested. You can change that to whatever you want. You can keep all that to 0, or you can increase the, does that make sense? Okay. So in the table again, we have the number tested for each age sex class, right? And then this is something I was referring to. I kept on adding stuff as things became clear. So some of this might not be necessary, but I at some point realized I want to know how many CWD infected animals were actually harvested. Not all of them are tested, right? A proportion of the harvest is tested. So then we have the next 6 columns which document how many of the total infected animals were actually harvested, right? So notice you have, where is it? So CWD hunted deer, none of the CWD infected deer were hunted in the first year. Of course, we just had one walk into that thing. So that particular yearling being harvested, the chances are very low. CWD tested deer are 0. Now, this detection probability is something we come to, and then things start becoming a bit problematic in terms of understanding it. Peter and I were talking about it yesterday or this morning about it. So just to be sure, detection probability, as the definition goes, is the, and I have written it down there, but I know the Zoom participants cannot see it, is the probability of detecting chronic wasting disease or any disease, given that the disease is present at a hypothesized prevalence. It's not just the confidence of detecting CWD or any disease. It is the confidence of detecting disease if it occurs at or above a certain prevalence, right? And that gets us into situations which make all of this surveillance really murky. You look at the papers that have been published on CWD surveillance. For about 25 years, we have these papers, and they all talk about hypergeometric distribution. Later on, they started talking about weighted surveillance and all of that, right? Keep in mind, and I tried drawing that here, it probably will be kind of confusing still because there is a lot of articulation that is needed with this thing. But I'll try and just put this out for you. This is no new knowledge. I'm just trying to put this out. So you remember we have those 2x2 tables for disease positive, disease negative, test positive, test negative. Now this is not a test. We are out of that. We are not talking of sensitivity or specificity here. What we are saying is for a given area of interest, the area either has disease or it does not have disease, right? And you will either detect disease or you will not detect disease. It's not about sensitivity specific, it's just about the outcome, right? Keep in mind, this will never happen. If disease is not there, there is no way of detecting disease. I'm not talking about the test outcome. It's like, given that you have a 100% sensitive, 100% specific test, and you sample all the animals. If the disease is not there, you are never going to detect the disease, right? Does that make sense, right? If the disease is not there, right, and you do not detect, this is what will always happen. If it's not there, you are not going to detect it. But the problem arises with low prevalence of chronic wasting disease or any other diseases. When you are in this part of the 2x2 table, the disease is present at a very low prevalence. So you detecting the disease is very difficult. Because of the required sample sizes and all of that, you almost always do not detect the disease at this point of time when you have a low prevalence. As the prevalence changes, or in some cases, as your sampling intensity increases, you come into this part where you want to be. Disease present, disease is detected. Now, the real world problem is how do you differentiate between this and this? The outcome is you don't detect disease, right? And so no matter what you do, when the disease is not present, you never know in the real world unless and until you test each and every animal in the population. You cannot be confident that the disease is absent. So how do you differentiate between these two is the key point of surveillance, especially for a disease like chronic wasting disease, right? And that is where this concept of detection probability, was really being discussed. And detection probability does not mean just that whether you are able to detect the disease or not. It always comes with a caveat that the ability to detect disease, which means at least one positive in the animals that you test, at least one positive, given that the prevalence is 1%, or given that the prevalence is 0.5%. So that given that concept is very important. Otherwise, if you use detection probabilities, it makes no sense. You cannot just increase your detection probability without having the hypothesized prevalence stuff. And much of the confusion actually centers around this misunderstanding. So I will not go into those details. I am going to bring up the surveillance model later on. But what I want to point out is this does document detection probability based on if CWD positive animal was tested, not just harvested, but tested in that year, right? So notice there is the detection probability of 1. It can only be 0 or 1 in the model, okay? It is 1 because 5 CWD positive deer were hunted in that year, in year 8, right? Or year 7, and only 1 of that was tested, which of course was positive, because in the model right now, the test is 100% specific, 100% sensitive. You can also adjust that, right? You can put in a specificity sensitivity so that CWD tested does not equate to detection probability of 1. You will miss on some of those. That's very easy to do. Does that make sense? Right? I just want you to look at what all is being documented right now. Observed prevalence. And again, as I said, post column AD, we are getting into kind of concepts that are really misunderstood, that are really murky and all of that. So I don't like the term prevalence to be used when we are talking of like diseases like CWD or African swine fever, all of that, because it essentially is a snapshot, right? Also keep in mind that I am here saying observed prevalence, so it is actually the sample prevalence. How many were tested? Out of that, how many were positive? That is what is being documented here, right? We can very easily know the true prevalence. We, of course, know that because you know the total number of infected in the population for that year, and you know the total population size. So true prevalence is actually written on the interface. If you look at the interface, which is this, it should have all of this here. So number of CWD infected deer, the true prevalence of CWD for that time step, and I'm going to talk about this. I think that is the most interesting part, but I will come to that in a moment. Does this all make sense? Any comment, any question, please let me know. There are 2 comments, chat. Okay. So back to the Excel sheet. Now this, as I said, I have, probably Barb, if she is online, she remembers that. So when we were in the initial stages of putting the CWD infection model together, a point came up during one of the meetings where they were interested in the geographic spread. Can we actually look at the pattern of geographic spread of this disease? So what we are documenting right now there as CWD area is the patch where CWD was introduced, right? The orange colored patch that you saw on your interface. And every year when this is documented, it counts the number of patches that has CWD positive deer on it. And then it finds the max distance. So you have the area and you also have max distance from your initial seeding point. And I will come to an important metric that kind of gives us some really interesting insights. I haven't been able to kind of build that any further, but I think there is a promise in there, and I will in a bit come to that point. So area of chronic wasting disease, geographic area, number of patches that have CWD on it, max distance from the point of introduction, right? We can actually now calculate kilometers per year. Yeah. Yes, that is miles. Yep. And so you are referring to the row after the first row, wherein you have CWD area of 1 and the max distance of 2. So right, and not the deer agents, but in NetLogo, what happened, there is like, it has its peculiarities. So a patch, which is 1 square mile, the distance is measured from the center of the patch. So the patch next to it, the patch next to that patch, the center of that patch is exactly 1 mile, right? So you have one patch, an adjoining patch, and the distance between these two is 1 mile. Right, right. So the one patch is the patch on which at that point of time you have an infected deer, right? So what might have happened here, if you notice, where is that? Yeah, so we started with one infected deer, right? Where is that one? It should be back here. Total CWD here. See, you started with one back. What changed? You had an yearling male walk in. That was the only deer documented in year 1, right? In year 2, you don't have that yearling male anymore, but you have an adult female, right? And you have an adult male infected. So this one probably died, right? So it's still counting the distance from the original patch, the orange patch, where it was. But now you have 2 infected deer, and so it's counting the distance to the farthest patch, and then they are both neighboring patches, so it came to 1. So you are right, but these are idiosyncrasies of NetLogo, like, yeah. But I think, and I'm going to get at that important metric here, but this is something I think is really exciting. I briefly mentioned it to you this morning, but this is actually documenting each and every transmission occurring in the model, whether that deer remains alive. So let me back up and give you an example. The information is written every year in a particular time step. Let's say it's written in Jan of every year in the model, right? So in Jan, it looks at all the infected deer present in Jan, right? And then fills up all of those columns. But suppose it so happens that from Jan to December, you get like 10 new infections and 9 of them are harvested. So the next time you reach January, you just have 1 new infection. 9 of those were infected, but they were all harvested, right? So with cumulative transmission, anytime a susceptible deer gets infected, it is added to the cumulative transmission counter. Whether it dies or whatever happens, we still have the cumulative transmission counter. And you might notice that by the end of 15th year, you had a total of 292 transmissions occurring in the model. That does not mean all of those 292 were present when prevalence was calculated in the final year, right? It just calculated the surviving deer. That can mislead us because, in a particular year, due to stochasticity, a lot of infected deer, susceptible deer that became infected, were harvested. and your prevalence from previous year did not change a lot because you are just taking snapshots if you just consider prevalence. With cumulative transmission, you actually know the exact pressure, or let me put it in a better way, per capita rate of getting infected. And that is the force of infection in the technical terminology. The rate at which susceptible hosts get infected is the force of infection. And that is a standalone metric, much better than prevalence, because you actually capture all the transmission happening in your population of interest. And any intervention you do and compare it with no intervention or status quo, you can actually see how many new infections you have prevented or not prevented, right? And because this is a stochastic model, I think this is particularly important. Then if you use the targeted culling option, this will document how many were post-season targeted culling. You can actually document the effect of that and how many of the target culled individuals were positive. So we can, if we have time, talk about targeted culling in detail. But the idea behind sharpshooting or post-season targeted culling is that you go into an area where you have detected CWD and you want to reduce the density around that detection so that the transmission is prevented or slows down. That is the idea, right? I know most wildlife agency people do not agree with me on this, but yet I'm going to just put out this point there. I will also highlight that when we look at the CWD trajectory coming out of this model. But keep in mind, the pattern in which CWD spreads, it does not, no disease spreads on a level playing field. It does not happen, this patch is infected, now you have the next patch, neighboring patches infected, then you have the, it doesn't, it makes those jumps, right? So it ends up as in you have a foci of infection, and then you have sparks somewhere out. With the post-season targeted culling, what most agencies assume that the case that you have detected belongs to a cluster of cases, which is not the case always. Sometimes it is on its own. So if you go around every detection and start doing targeted culling, you will end up with a lot more negative deer removed from the population. I am not saying this. Look at data from any state. Well, except the states which has had like core areas where the prevalence of CWD is high. But all states that have detected CWD in the last 4 or 5 years, look at their post-season targeted culling data, and you will see it's all, most of them are zero prevalence, or you have find 1 or 2 in 70 deer removed from that. So it does not actually, it does reduce the density, but it does not really change the probability of transmission the way you will expect it to happen in an endemic zone. And we are going to talk about that later on, but here you can actually look at the impact of targeted culling. So is that clear? That is what is all written from the infection dynamics model, right? Any questions from the Zoom participants? If not, I am just going to go back, and I am hoping that at least some of you start the run with 100 iterations for the CWD model, so that you can have your own file you can look at later on. But if you don't want to do that, it's all fine. I can briefly, it's the same thing, same procedure we followed. So you will open the CWD dynamics model, which is this. Except for you two, others have done the changes wherein we added La Porte here, wherein we went into the code and changed it to 15 years. Remember, just in the previous session. And then we also put this La Porte, access to La Porte output file here. Those were the 3 changes we did in this model. So anyone interested can go to the interface. Before doing that, go into the results folder of CWD, OVCWD, and delete the results file. So you will not overwrite it, right? come to this part. Yeah. Okay. Come to this part, and you have all of this in place. You will select tools, go to behavior space, new experiment. I would just call it LP 100. So 100 iterations, right? You will then come to repetitions and type 100 here. It should be 1, type 100 there. So it's going to do 100 iterations. Delete count turtles. We do not want that. Once you are done with all of that, you will go down and say, okay. It should open this file with your new experiment in there. When you say run, it should open another window wherein you should have nothing in the spreadsheet output, nothing in the table output, unchecked update view, unchecked update plots, no, nothing there. Just change this to 1. If you make that 1 and press OK, your behavior space experiment should start, and it should stop only after 100 iterations are done. Does that make sense? Sure, sure. Oh, sorry. Okay, so everything else is there. You just go to tools. In your case, it is settings. Oh, it's still tools. Okay. In tools, go down to behavior space, click on that. Right? I would always create a new experiment. Okay. Give it a name that is easy to understand. So LP 100 is what I would say, but you can be as creative as you want and put some name there. If that is done in repetitions, it should be 100. We did 10 last time for the OVPOP model, but here, because there is a lot of things are happening in terms of infection and harvest and all of that, using some basic statistical approaches, we have determined that we need a minimum of 100 runs to have results that are reproducible, right? Delete everything in here which says count turtles. We don't want anything. Delete that. If you are done with that, 100. Delete count turtles and click OK. Then you should see this with your new experiment name after you click OK. If you see that, then that has to be selected and you should say run. Once you do that, it will open another window where you have everything unchecked, everything empty, but this should say 1. Simultaneous runs 1, not 4 or 16 or 18 or 20. And then you press OK, and it should run for 100 iterations. Okay, great. So I am just going to go back. Keep it running, Peter, if you have it on. So we'll come back to it later. But I am going to come back and just discuss more about what you would see. Okay. So why is it that we do 100 iterations? This is one of the iterations, one of those 100 iterations. You are now familiar with what's happening here. A yellow patch means an infected deer is on there. The number represents number of infected at that point of time. An empty yellow patch means there was an infected deer which has been removed, right? Yeah. That is a great question. Yep. So I started with that. What we have put in right now is the prions remain infectious for 2 years. Okay. And Peter laughs at that, and I agree with him. So we can. It's very easy to change that. But the real objective here is to have that in and have environmental risk map in our population. Right. Could become infected. Exactly. And the orange patch represents the patch where your first dispersing deer, male deer came into. And so this is one pattern, right? But if you look at the next one, it might look something like this, and which I guess you had it, right? One of you had it where, it just died off. The infection. That is a process that was documented in 1980 by Anderson and May, seminal work. They call it stochastic fade out. It's not only for CWD, it's for any infectious disease. What it means is, despite having susceptible individuals, oftentimes a disease introduced in the population due to stochasticity, of demographic processes, it can just fade out. That is what you are seeing happening here, right? So these fade outs, we haven't coded it in the model, right? In our model, we don't say so many % of runs are fade out. This is an emerging property. So an infected deer, is harvested, and then there is no infection left. And so those runs probably would look like this. Or an infected deer dies because of the disease or naturally before passing it on to any other deer, and then it would look like that. Okay, so it's a combination of persistent and non-persistent outbreaks. This is very important. I'm going to come at that metric, which I find very intriguing. I would like to know if you find it too, but give me a minute here. So because you have two types of outbreaks, one that is persistent, the other which is not, you cannot just do 1 runs or 5 runs or 10 runs, right? We need to do a lot of runs to capture all that variation happening and then generate a statistical portrait. So we run 100 iterations using behavior space, and then we go to the same Shiny app that you guys have used. Okay, you have access to the Shiny app, you know the login and password. I have a file here for you on my GitHub repository, this link. You can right click it and download this file. This is the result of 100 iterations. Peter, you will get the same file after your runs are complete. But right now you can access that file. Okay. And do you want to share your screen again, Peter, after you are done? Not necessarily. No, you don't have to do anything there. You have to open the app that we used for our OVPOP output, the same app. Do you need the password again? I'll write it down. It is user 122 and *3. User 122 *3. Zoom participants, if you need the password again, please let us know. But you go to the app, log in, and before that, please download this file. This is the result of 100 iterations. Right click on this link, open in a new tab. It should open my GitHub repository with this raw file, and you can just, I think, do a right click and save as. Just make sure that you save as a CSV file. Put it on your desktop because you have to upload it on the app. Yeah. Let me know if there is any issue here. Okay. All good until here? Access to GitHub and all of that works? Okay, so you can scroll all the way down, not all the way, halfway down in the web page, and there should be a link for the Shiny app right around here. The Shiny OVCWD, again, right click and open. So now just that you know, because when you open the app, it will let you choose the file. So if it's on desktop, that's fine. Yeah, or wherever you prefer, yeah. Login is user 122 *3. And now just don't fill anything else. It's very straightforward now. You don't need to do anything on the sliders. Let me open this up as well. So open in the new tab. Okay, you have this, you log in. Forget about all the top part. You have to go to the OVCWD DI model output analysis. The window, the tab at the very bottom. Select your file and upload it. And I'm going to close this. I want you to share your screen. Yep, I'll stop sharing. And please give me your suggestions to make this more accessible, more meaningful, and all of that. I have been working in like kind of a, that's the reason I'm so excited to be in this room. Like all your suggestions to make this more accessible will help. And I know I have done a lot of small silly things in there which can be easily corrected. So please let me know. Right. So you have uploaded the file. You haven't your login isn't it working? User 122 *3. Oh, did I give you the wrong password or anything? No, no space. Ah, that's interesting. Oh yeah, okay. Everything is small. Yeah. Okay. The file is in there. Yep. And so go to the top and then click on OVCWD trajectories, the third there on the very top. There, yep. Okay, let's see if the file works or I fall flat on my face. It worked? Okay, good. Okay, so now I want you to understand that for these two plots, you can actually play with the y-axis to make the graph more reasonable. So bring it all the way to the left to 5,000, and then it will make some sense. Even here, you can bring it all the way to the left. No, there, yeah. So what you see on the screen here is these are all the persistent outbreaks, and these are all the non-persistent outbreaks, right? Um, Even looking at this plot will not make sense because you have a lot of variation there. So I converted this plot into a graph. So can you scroll up? Yeah. Again, now this axis, you can adjust it with the y-axis. The reason I have provided that y-axis adjustment is because some counties where you have a lot of CWD, especially when you look at a endemic phase, you need the y-axis to be really good. So you can go maybe all the way to like 0.15 or something. Yep. So what do you see here? It is the same plot converted into like summarized into a mean. And this is what is our CWD trajectory. Okay, we have CWD introduction in the first year. Okay, for about 10 years, the prevalence remains below 1%, which is all of this. And then you have it increasing. After 15 years, you have like about 5% CWD prevalence in this county. Now, I want you all to think for a, oh, I guess that thing I need to do it again. Sorry about that. I don't know if it bothers you. If not, it's fine, right? Okay. So, yeah. Very interesting. I don't know if folks have been.
So I always think this is super interesting. The first time I saw this back in November, whenever I visited Ani, in Minnesota and in the southeastern part of the state, for the last few years, it's always been around, I think, 1% or so. And I think that now we might be entering that transition phase. And so, Ani, can you talk a little bit about where have you witnessed this, and
sure.
That helps us better understand the model.
Sure. So that is the next part that is on my page. But before you do that, I realized this way later while interacting with wildlife agencies and a student I am advising at Indiana, is they were stuck up on this plot. And the wildlife biologists kept on saying, this doesn't make sense. In 15 years, I have 5% prevalence. This is not what the model is saying. This is something you need to understand. And I am sorry, it is my fault, but you can, oh, sorry, I can open up on the web page now. So you have seen that the file works, right? Okay, so let me just pull that up real quick. Yes, please. Okay, give me a minute to just bring that up here and we will take it from there. And I need to share again. Okay, we'll use a different region this time. This has a lot of issues, this plot, right? What do you see when you look at it? Like, I haven't given you any information other than what is out there, right? First of all, we are able to implement really nuanced scenarios. So for this particular county, I think it's 2 counties together in northern Missouri, okay? They had a mass die-off of EHD in 2012. And all of this data is based on those years. So we were able to bring in the EHD die-off, okay? APR was implemented up until 2012, and then they discontinued it. So we have that change incorporated in here, right? And if you look at the plot, this is telling you that in 25 years, you have like a 50% prevalence. That does not make any sense to any biologist, any white-tailed deer person, just doesn't make sense. And one of the reasons why it doesn't make sense is because this is not a standalone output. Okay, if you interpret this as a standalone output, you are going to think of it in the same way as the person who kind of was not able to agree with this. And I'll tell you what you have to look this, what you have to pair this with. So this output, which you saw on the Shiny app, slightly different, but this is another concept that I think ties in with the stochastic fade out. Okay, there was a paper, and I have given you the link to that paper. I also sent you the paper which you wanted me to share with the group. Okay, so this term was coined by Silk, in 2018, 2021. In their paper, not on CWD, they talked about demographic competence. This is a great term. I was calling this outbreak probability, but I like this very much. And this paper was published just after we published the CWD modeling paper, so I was not able to use the term demographic competence, but I make it a point to use it now. What is demographic competence? It is the ability of a population, a host population, to sustain an infectious disease out. It's plain and simple, right? So I created a waffle plot based on the very same runs that you did, okay? Remember, the blue squares in this plot, this particular plot, are the iterations where CWD persisted. The gray squares are where CWD did not persist. There was a fade out, right? So very simply put, demographic competence is 0.51 or 51% for Linn and Macon County. I was doing a workshop with the MDC people. This was 3 years ago. I did not know about APR. They hadn't provided me that data. We just had the harvest data. APR is antler point restriction. So it's a harvest strategy that most agencies, most hunters wanted, with the idea that if you protect yearling males from harvest, they grow into adults with larger antlers and you have more antler deer in the population, right? So it's a very popular strategy with hunters. And so agencies, many places go with that strategy, wherein you are not allowed to hunt an yearling male or something like that, right? So I did not know about it. During the workshop, Lonnie Hansen was there, and he is I have been working with him for a long time while developing the model. I presented this plot, and I made a comment, like offhand comment saying, this is weird. No other county that I have analyzed with these models has a demographic competence of more than 35, 36, 37. I have rechecked this several times. Why is it like 50? It's high. 51% is a high, which means a disease introduced in Linn-Macon County has a higher chance of getting established. And it went off. You know, we did the workshop, we went out for beer or something like that. And it came up, he said, has it anything to do with APR? Because this area was under APR for a long time. And it is exactly that. So APR, you have a lot more yearlings that can disperse. I'm not saying APR is bad. I'm just saying, like, for all the, we did that for Michigan as well. We evaluated APR strategies, and there are a few plots down there. What you can see here is the demographic competence is high. Again, this is an emergent property. I did not code it. The data went into the model and gave us a higher demographic competence. Going back to the trajectory that we were looking at, the trajectory does not say 25 years later you will have 50% prevalence. No, what it is saying is there is a 50% chance that introduced CWD will not persist. Of those which persist, if you plot a trajectory, a mean trajectory, it will look like that. But that still doesn't give us all the information we want. So I changed the waffle plot to a better plot now. And that is what you see on the Shiny app. But let me see if I have it here. Okay, so now I changed the waffle plot to look like this. And let me make it smaller. Okay, tell me if this makes better sense. Now, just remember, the blue squares are no outbreak squares, okay? Maybe I need to keep the same color, strategy. But now we want more blue squares, which is no outbreak. And so this is for a different county, but then there. So what is the demographic competence for this county? It is 55%, because non-blue squares is the demographic competence when CWD persists. The blue squares is no outbreak. That is what we or wildlife managers want. So what I'm saying is, if we can evaluate multiple harvest strategies, and for each harvest strategy, we create a waffle plot, the strategy that results into more bluer squares is the one that we want, right? And this brings us to a very important, again, point, and please stop me if I am kind of like, going here and there with making no sense or anything, but this is all connected. I really want to talk about two very important metrics, and then we can kind of, go back into the discussion, okay? I'll make the first point before getting into the important metric, but that is what, again, the last 3 years and this is where the point that you wanted me to kind of talk about. I think this is something that most wildlife agencies have failed to incorporate into their response plans. And I have done this kind of arbitrarily, but I think it makes sense, and you can, of course, comment on this. But this kind of a trajectory that you see, right? This is derived from, this emerges from our model. If you go and look at real world data, which is, I'm just giving you one example. Okay, need to make that bigger. You must have seen this, but let me see. Okay. So what you see here is for Pennsylvania, West Virginia, Wisconsin. There are several other studies which use field data or modeling data to create such trajectories. Notice that CWD prevalence trajectories for these states and for other states follow the same pattern. You have an early part where the detection remains low, no matter where it is. And then you have this kind of exponential growth. This is from detection. Detection almost always is never when it is introduced. So what I'm trying to say is this kind of goes all the way back here. So you have a long period where it remains very low. And then you have like this trajectory going up. This is the reason why I truly think if you do not have a phase-specific strategy or a phase-specific understanding, you are just wasting your resources and the valuable time. I think I am being blunt here, but this is a major issue I have seen over the last decade. There is a cut-paste approach of developing a response plan based on what 10 states before them have done, right? Going back, what I am proposing here is almost about 15 years since introduction. We never know when the introduction happened, right? But for the first 13 to 15 years, when the prevalence, the true prevalence, okay, remains under 1%, let us call that a pre-establishment phase. The disease is at a very low prevalence, and we can safely say that it has not become established yet, okay? Post 3% prevalence, so this is 3%, post 3% prevalence, you have an endemic phase. Once you are in there, there is no way of eliminating CW. It does not go back. It will be there forever, okay? This is what I have completely made up, and I am open to your suggestions and everything. I am calling this phase a transition phase, right? A transition phase where prevalence changes from 1% to 3%. Again, this is all model-based. There is no validation from real world. So keep that in mind. But I'm going to bring up those two metrics that I think have really valuable insight for all of us. But my understanding here is in this transition phase, we have the ability to delay the establishment and spread of chronic wasting disease. We do not have that ability in the endemic phase. Point number one. So you have a small window when you can actually delay you can do something meaningful. I have a further kind of plot to show that. So how did I come up with these phases, right? And I have to then open up a different
So this is super interesting. I was talking to Tiffany a little bit ago, like, you know, Minnesota has one of the, what would you say, Tiffany, the most aggressive approaches, right? Most proactive, aggressive approaches to cull, yeah, for management for CWD. So it's gonna be really interesting to see what the next 5 to 10 years brings, obviously.
New York was able to get rid of it, right? Oh, because New York is the only place we know of that's had it show up and they got rid of it, in theory, right? Okay.
Okay.
Because I'm wondering where we were on these curves, what things looked like there.
Right. It was the part of the model where the individuals. So yeah, we were talking about New York, right?
Yeah. So I will just, you know, I will not, it will take some time to bring out those plots, but I am going to talk about two model derived metrics, which I think have a lot of insights that we can use. Again, we cannot determine these in the real world, but just hear me out. So you remember on that Shiny app, you had two plots. One showed persistent iterations and one showed non-persistent iterations. If you want, you can bring that up again here because that will make sense if people can see that. Or is it gone now? Yeah, okay. So I'll try and find that plot here because there are too many plots that I want to talk about, but I think I need to keep it limited to, you know, what we are discussing right now. So let me see. I think the persistent and non-persistent plots, that is the important part of all of this. But I can explain maybe, yeah. Okay, so going back into the model output for multiple counties that we ran this CWD dynamics model for, what I found out is analyzing the non-persistent iterations, right? So in this case, there are 45 non-persistent iterations. 45 times the outbreak petered away, did not persist, right? I kind of analyzed all of that. I found two very important, points. The geographic spread, when it is under 40 miles, after year 5 or after year 7, before year 10, the outbreak does not persist. And the number of infections, the number of infected deer in the landscape, if they remain below 25 or 30, there is a good chance that the outbreak will fade out. So we now have metrics. One is 40 kilometers or 40 square kilometers, and the other is 25 infected deer, right? These are the thresholds. If these two thresholds are crossed, you have a high chance of having a persistent outbreak. Now, again, I am not saying that you can go out in the real world and count all this, because that is uncountable, right? But at least in a kind of a theoretical kind of way, we now have two thresholds, which if we design interventions and look at how these interventions affect the number of infected deer in the first 10 years and the geographic spread in the first 10 years, we have something to justify which intervention is better than the other one, rather than just looking at number of infected deer, right? And this can be refined further if more heads are put together and if we have a lot more statistical prowess to look at like thousands of non-persisting iterations, right? Different landscapes and different geographical features. We can look at, can we have something to compare between set of interventions, is one point I'm trying to make. That brought us to the phase specific approach, right? And this is the second point, and then I will let you guys speak, but just hear me out for this one very important piece that we have here. I have other stuff which we can talk, but I think right now I want to focus on the phase specific strategies. There will be some criticism for what I am going to say, but I am willing to take that. I am willing to, yeah. How do I make this bigger? Is it becoming? Yeah, but it's kind of, okay. I think this is good. Okay. So, yeah. You remember this trajectory, right? This is the same trajectory we talked about, right? First 13 years, you have under 1%. It's exactly the same. I just brought it here. What I added to it is we also have a surveillance model. That's the one we published early on. And what that model does is it gives you detection probability for a given sample size and a given prevalence. So very quickly, this line here that you see, what color is that? I don't know what it is called. Turquoise. Turquoise. Okay, so the turquoise line that you see down here is based on a hypergeometric distribution. It's the sample size tables that all agencies use to determine sample size, right? So the main two assumptions underpinning the hypergeometric distribution are, one, that the disease is randomly distributed in the population, and two, the sampling is random. Hunter harvest is not random sampling. Okay, keep that in mind. So if you are willing to violate both these very crucial assumptions, you are good with this line. So at a very low prevalence, you would need about 600 samples, very low prevalence, to detect CWD, right? As your prevalence kind of increases to nearly 1%, you could do with 300. Go and check the sample size table. It will give you 293, right? For 1% prevalence. in like, this was done for like a 25,000 strong deer population or 40,000, you just need 293 samples to be 95% confident that you will detect CWD if it is present at 1% prevalence. But these assumptions are all violated in the real world, right? So I ran the surveillance model with modified assumptions that the disease is clustered and sampling is non-random. That is the top line. So it just tells you how many samples you require for the same prevalence. And this is not possible. Like 3,000 samples from one county is not logistic. You can't do that. Norway did that. But it's not possible, for most of the areas that we have. What this tells us is in the pre-establishment phase, active surveillance does not work, will not work. It has never worked. You will detect it by chance, by passive surveillance. Arkansas detected it in 2016 because they had a roadkill come up positive and they looked around the area. That's the same case like New York. Yeah.
We were talking a little bit about this. Here in Minnesota, it was it Grand Rapids? That case was like unexpected, right? Like, I mean, this was a deer that was sick in someone's front yard, and all of a sudden it was this, whoa, we have CWD up here in the north. So, yeah.
And so, like, we can still come back to some of the issues with this, but let us for now agree that in the pre-establishment phase, when the prevalence is very low, detecting it with hunter-harvest sampling, you cannot reach the intensity that is necessary, and it will remain undetected. Now, notice the reason of this transition phase is even in our realistic model, the prevalence has changed from 1 to 3. You see a drop down in sample size. And the simple reason why this happens is in the surveillance model that we have, even if you cluster your cases, at a certain prevalence, there are so many cases that they are distributed in the landscape and your ability to detect them increases. That is what is happening between 1 and 3%. This is much more doable, like, about 1,000 to 200 samples to be 95% confident that the disease is between 1 and 3%. In my mind, that gives you a window. So you don't have to do that every year for every county. You can have a strategy where once in a while, once in 5 years, if you sample with an intensity that will detect confidently in this window, right? It is a 5-year window, at least from the modeling point of view. You can do intelligent sampling rather than exhausting the agency, kind of exhausting itself, doing a lot of 30,000 samples every year, and then the resources and the energy dries up. One point is that for the transition phase. I don't want to talk about the endemic phase because that is something very, very grave, right? Final point is this table. And this is where I'll stop and we can have a lot more discussion also from the people there. But again, help me out, okay? I now have added another phase, which is pre-introduction phase. And that brings us back to the table I was trying to draw here, where the disease is not present in the population of interest. You and I do not know that. But just assume it is not present. So any amount of sampling you do in that is not going to give you any information, right? Remember, the lower 2 points here, where disease is present but not detectable, or disease is not present, hence not detectable. There is no way to differentiate between that. Unless and until you sample all the 25,000 deer here, you are not going to get any valuable information. I can go into the details, like there is one rule of thumb, I think 3/n. It's used by field epidemiologists. So you sample 50 individuals. All of them test negative. So 0 sample prevalence. You can use the rule of thumb. What did I say? 3/50. What is 3/50? Does anyone have a calculator? 3 divided by 50. Yeah. 0.06. So you can be 95% confident that the disease is not present above 6% prevalence. is what that zero sampling tells you, right? So otherwise, there is no information in a zero sample prevalence, right? You test 10 animals, none of them are positive. Does it mean there is no disease in the population? No, it is 3 upon 10, which is 1 upon 3 into 3. So 1 upon 3. So you are 95% confident that the disease prevalence is under 33%. How does that help you? It does not help you, right? Under 33% doesn't make any sense. So what I'm trying to say, backing back here, people jump at me when I say there is no utility of surveillance and it is not sustainable. I am not saying don't do surveillance. I am saying plan your surveillance in a manner that if the disease is present between 1 and 3% prevalence, you will detect it. If not, the only thing you know is once in 5 years, I sample county X for the transition phase. So I need a lot more samples than normal sampling, but that is only once in 5 years. If it is out of that, then I am confident that the disease is not prevalent above 1% prevalence, right? And I can look at other counties. That is the reason I am saying a general surveillance of the state, or as we were talking, there are 2,000 counties in the US. 400 of them have detected CWD. 1,600 haven't detected it yet. Doesn't mean it is not there, right? So what do? Do you go in those 1,600 counties and just keep sampling until finally you detect it when you are in the endemic phase? That is waste of resources. You need to be intelligent with your surveillance. The same thing applies for pre-establishment phase here, because the value of surveillance is questionable here. It's because of this sample size requirement, right? It is definitely not sustainable. Ask Michigan, ask Missouri, 30,000 samples every year. Talk with their people who do that work. Ask Barb. In the transition phase where you see green, that is the best way to use your resources, where you can be very focused on detecting it if it has a slightly higher prevalence. But if you fail to detect it, you can let that region be on your kind of second priority, right? And that is definitely sustainable if we do it in a coordinated fashion. Here, it is sustainable, but this is an entirely different problem. This brings us up to what our management should look like. For these two phases, for those 1,600 counties, we need to have preemptive strategies. Assuming the disease is there at low prevalence, what can we do to increase the number of blue squares? So if the disease arrives, we delay its establishment. If the disease is there, we delay its spread. We don't know whether it is there, but our strategies are to bring down the demographic competence, which we can measure, right? And your surveillance should be focused on the transition phase, where you can intelligently sample your state so that it's not just focused on where you find one case, but more important, I think, is counties where you haven't detected it yet. Arkansas, the first month in 2016, they found 100 infected deer. Until then, they were doing their surveillance, but they didn't find any, right? So that infected deer, it was probably luck by chance that they found the first case in a cluster. Other states, New York, I just question their claim that they were able to, they didn't do anything differently. It was either a stochastic fade out or it was a spark that they detected. If their strategy worked, what is that strategy? Who knows? I haven't seen it published anywhere. And finally, again, I think, weighted surveillance, which is being talked about, very much these days. Keep in mind, it works here. It doesn't work here. In the pre-establishment phase, weighted surveillance does not work. If you assume you are in like more than 1%, more than 3% phase, it will work as they claim. It's of no use to detect the disease when it is here. So bringing your sample size down happens here naturally. You see that sample size requirement does go down in the endemic phase or close to endemic phase. What we want to do is detect promptly. And if you want to detect it promptly, I think you need a strategy that captures it in a known kind of like, you have the certainty of capturing it, even though it seems a higher prevalence, but you don't wait for that to happen. You do preemptive strategies in those 1600. Okay, I think, I do have some other stuff, but let's kind of open it up. Yeah.
Okay, so my question is, when you've got management agencies that you might want to present this to, and I'm assuming have presented this to, I imagine the next question would be, well, how do we estimate how much of this pre-introduction effort we need to put in? How many years? Do you have estimates for that?
Right. It's there, sorry. So we tried answering those questions in here, okay, which is, I will summarize it for you, but we have tried answering those questions. Sorry, it's like just going up and down, I know. Yep. Here, in this paper, getting in front of chronic wasting disease, model-informed proactive or preemptive practices, for this chronic wasting disease. What we really want to say is, first point, CWD, as like other infectious diseases of wild animals or free-ranging, free-roaming hosts, does not spread on a level playing field. So what you see in Missouri might not be the case in Minnesota. That doesn't happen. You cannot just say that, oh, this spreads at 12 miles a year in Missouri. It doesn't mean it will happen the same way in Minnesota. So you need to first understand, have some way of documenting or incorporating that into your decision-making process. Secondly, the demographic characteristics is way different. Like, I just repeatedly make this point. I know, the APR thing that we found out with the Linn and Macon County, it was not put into the model. It was just the data reflecting that, right? So it was because the demographic structure of the particular population was in such a way that it kind of accelerated the spread of this. It didn't happen with Montcalm County, Michigan. It didn't happen with several other counties in Missouri and Michigan that we always ended up with, for the status quo scenario, we ended up with 0.33 to 0.37, 0.38 demographic competence. So very few iterations actually resulted into establishment, unlike Linn and Macon County. And then, as you said, we said, well, let's see what happens if we remove APR, if APR wasn't implemented, right? This is the beauty of virtual experimentation. So we did that for Missouri and we did it for Michigan. And this is what you see with Linn and Macon County. I hope I have the plot there, but should be a plot which shows, okay. That is the force of infection. But we have a plot somewhere here which compares APR in those two counties, in two scenarios of APR present and APR removed. But I have something else for you. Maybe we can actually look at the dashboard here. And all of you have access to this. Anybody can open, and look at it. But here's the work that we did for Michigan. Okay, there is some information here about the modeling framework and all of that, but this is how we develop the scenarios. But look at the APR scenario, right? So very quickly, APR is implemented in Montcalm County, Michigan, okay, in the pre-establishment phase, right? So when the prevalence was under 1%. The blue line is the baseline, no APR. The red line is when APR was implemented in the pre-establishment phase, okay? It was implemented in year 13, when actually the prevalence was very close to 1%. And statistically, I guess you have it's statistically different, these two trajectories. You can go into the details and look at it. But then, because we want a phase-specific approach, what if it was introduced later, when the disease was endemic? You still have a statistically different, significantly different trajectory. So further, what if it was introduced before CWD was introduced? You still see a lot more CWD in the landscape than the baseline scenario. So let us go to the other extreme and instead of APR, let's increase the harvest of yearling males, right? And so here we have a slight increase in yearling male harvest, but here you have like slightly more, 10% increase in the yearling harvest, and here it is 15%, and then you see less CWD in the landscape.
So I feel like this is the perfect storm because over centuries, we've converged on prizing the old, mature bucks, right? So, you know, there's that aspect of hunting that this is like the perfect storm, right? You have a disease where.
And I think, again, I'm being very naive about this, but when I developed this dashboard, and there is another Shiny app where the user can open it on a cell phone and play with the slider to look at the impact. So these are all model runs that I did and stored the outputs in GitHub. And the Shiny app just accesses the relevant model outputs, the files that you have. So I did like 1,000 runs, put all of those runs on GitHub, and the Shiny app, based on what value you select, accesses the necessary file. So the user does not have to run the model, but everything is open access. So they can go in and look at that thing. So the Shiny app, actually, I thought, no agency person agrees with me, but I think it is a way of meaningfully involving or engaging stakeholders. If the hunter lobby is not agreeing with you, bring them to the table. We will design a Shiny app, let them see that, and they say, no, this is wrong. This doesn't happen. Let's change those assumptions in the model, rerun the model, and bring the results back to them. But they think I am too naive, and I might be so. So, you know, I do think stakeholder engagement is important, but you know, that's.
Do you think for all the, you know, across the US, all those estimates of, you know, prevalence based on hunter harvest, are we just vastly underestimating CWD prevalence across the lower 48?
I think, and again, you know, maybe correct me if I am kind of overstating this, I think the spread is vastly underestimated, but the local prevalences in many cases are overestimated. And that again goes back to the phases and the sample prevalence. So you cannot connect the sample prevalence to the population prevalence as all the agencies do, right? One county, you have 100 samples and 10 of them are positive. So your sample prevalence is 10%. What most agencies do in their reports is say this county has a 10% prevalence. That is not correct. It doesn't connect that way because these samples are not representative. They are not random samples. They have a lot of, like, as you said, there is a hunter choice, then not all of them are tested, right? So you really need a model-based tool to convert the sample prevalence into some sort of the true prevalence or estimate of true prevalence. And that is what I guess, you know, we are hoping to work on with another model if we have time, or if not, we can talk about it later. But I think the spread is vastly underestimated. All those states that I talked about earlier on when they didn't have CWD, I made a comment like, you are saying you don't have CWD, but I say you haven't detected it yet. And in the next 2 years, you have it detected, right? So my point is 1,600 counties undetected does not mean that they don't have CWD, but local prevalence, at least in the endemic areas, I think it's very important to make those kind of, you know, prevalence estimates, putting them out, because sample prevalence does not directly connect to true population prevalence.
We talked a little bit about Mississippi, and there was a county where it was not detected, just like you said, and work that Stuart did with Mississippi State on swabbing, or I think it was the scrapes, right? It was the scrapes, and they had detected CWD in the soil or the licking branch in a particular county, unknown to circulate at the time, and it took over a year, I think, before
Right.
confirmed,
Before it was
right? So
confirmed.
as far, I mean, this gets into what we've been talking about, about other methods to
Right.
conduct surveillance, right?
Right. Agreed. Any other comment from the Zoom participants from here? But if not, I just have a couple of other scenarios I just want to show you so that it might result into you thinking of some creative scenarios. And there are many, I just selected some for you. So I'll go back. This app, you can access it anytime. It's again, this is here. This was created for MI, Michigan DNR, and this is, they suddenly lost interest in this app. They were very excited, but I think there was change in, whatever. So what you have is just a select a county, and there is this one slider, right? Right now, what you see is a baseline CWD trajectory, and you have the opportunity to bring down the APR, right? And look at how it impacts this. You can also say when it was introduced, right? Or you can, this was a scenario they suggested. What if there was like a compensatory increase in antlerless harvest? I think you need to be here. Yeah. So you see, you can get as nuanced as you want. One of the query was, okay, APR results into increase in CWD. You are protecting yearling males. What if, along with APR, we ask the hunters to harvest more does? So you compensatory increase the doe harvest. So we tried that. That is the green one, right? It doesn't make any difference. You still see a lot of CWD compared to the baseline. What I'm trying to say is this is not the final truth. This is just doing a what-if analysis based on what the managers think makes sense, okay? Finally, I just wanted to give you a brief idea of this study. And I know this is too dense here right now. I'll try and just explain what you see in this table, okay? So these are the different scenarios, right? The baseline scenario is the status quo, done nothing, okay? With what you see here, and I have lost my ability to come up with names for scenarios, for state variables, for whatever. So this is very kind of like, this is pre-establishment, low intensity, all age classes in 9 square mile area. That is the general logic. So disease is less than 1% prevalence, okay? As a part of your targeted culling strategy, you decide to cull all age class deer, whatever you encounter in a 9 square mile area, okay, at a low intensity, which is 2 per square mile. If you do that, and then this is medium intensity is 5 per square mile. This is like 10 per square mile. Now you get into B is antlered deer, just the antlered ones, and D is antlerless harvest, right? So the same scenarios. What I want you to focus on is how do we compare between these different scenarios, okay? There is a lot of variance because of the stochasticity. So deer removed by targeted culling, all deer that are removed, you see a lot of range. Like look at this, at the high intensity one, like you have to understand that, like, because there is stochasticity, which deer is detected during a particular model run results into where it will be implemented. So keeping that, I want everything to be open. I could have just presented this. It does not make sense, right? So here we have the true prevalence in year 15 of all these model runs, the baseline being 6%. You have 50% reduction in one of these scenarios, right? And this scenario. So pre-establishment phase CWD, high intensity, which is 10 deer per square mile in a 9 square mile radius, all age classes, and only antlerless age classes. For these two scenarios, you have like a 50% reduction in the true prevalence. At the same time, you have about 50% reduction in the CWD area, right? But keep in mind, like this comes with a lot of variance in there. And I try to summarize this in the agency report. So green is what you want, right? Red does not result into significant reduction. The yellow are okay, like you don't have 50% reduction, but you have somewhere between, about 25 to 40% reduction. But green is where you want to be. But what I haven't presented here, it is in the report, is if you look at the population decline with some of the high intensity, keep in mind, I just showed you the 9 square mile area. There is also a 25 square mile area around the detection. So the larger area you have for targeted culling, you end up into bringing the deer population down because you remove a lot of uninfected deer. Because we are in the pre-establishment phase, the results will look way different, and we have done that if you are into an endemic phase. Because, yeah.
Targeted culling strategy at density.
Okay.
And there are states.
Right.
They're not. I don't think that they are actually impacting the local density of deer, although that's what you hear from locals. Their approach is to target as close as possible to where positive deer have been harvested.
Agreed.
Those social groups.
Right.
And they're really, and I mean, we did hear from Barb, they're just going to kill as many deer as they can where the landowners give them permission.
Agreed.
It's not to reduce the density, but to remove as many contacts as possible. So I'm wondering, like, and I do hear from our colleagues, they want to know, is this effective?
Yeah.
That's a different approach than what we're hearing here. And I think it would be really interesting to try and figure out a way to incorporate that, because it's, like, when I think about targeted approach, that is targeted. Yep. This is more of a density dependent approach.
But you realize that they go into only detections. They don't do that everywhere. So any few detections.
Right, but 5 miles within a detection is a
large area.
That's beyond a normal deer's home range. So I think
you are exactly right. I think it has to be the local agency who design these scenarios. The 9 and 5 and 25 square mile is just not going to happen anywhere in North America, right? It will possibly be, but I just want all those agencies to understand one thing. And again, this is not like something I found out, but keep in mind, the phase is very important. So if you are in a pre-establishment phase with less than 1% prevalence, true prevalence in a given area, and you detect one positive, the underlying assumption on the part of agencies is that this one detected case is always a part of the cluster. That is why they want to go and work on the social group there with the idea that whoever is infected will die. But again, keep in mind that being a part of the cluster is not a case that will happen every time, or it happens very rarely in the pre-establishment phase. They make that sparks fly off, and you end up detecting you don't know whether it's a spark you have detected or it's a part of the cluster. Also, in the 1%, less than 1% prevalence scenario, the clusters are hardly like 2 or 3 infected deer, right? Yeah, you can, exactly. Agreed? Yep. Agreed. Right. Not based on simulation. Agreed.
That would be really, really cool next step.
Yeah, definitely. Yep. Yes.
Sorry, Zoom folks.
Well, hang on. Can Zoom folks, do we need to have the microphone, or did you hear Tiffany just now? So we don't understand how sensitive the microphones are in here. we'll just lean with the microphone for now.
So what type of radius are we talking here? We're talking 25 square miles. So that's like a 3-mile radius around.
So you detect a positive in one of these
pi r squared. So
no, it's not just
25.
No, it's that's a great point. Yeah, I'll
so that's like a.
I will show you what we do if we
like
come here, right? So again, if you detect it here, right? If you detect it in the orange square, a 1 square mile radius is just that patch, the same patch.
Okay.
A 5 square mile radius, again in NetLogo, like you can draw a circle, but what we do is we select the neighbors which are like, you know, at right angles, 1, 2, 3, 4, and this one patch. So it totally makes a 5 square mile area. For a 9 square mile area, we select all the, it's called a von Neumann neighborhood or something like that. So it selects like 1, 2, 3, 4, 5, 6, 7, 8, 9. That's a 9 square mile. And then the 25 one is like, again, it grows into that. So keep in mind.
So it's roughly close to it. I mean, it's like a radius.
It's like a radius, but yeah, it's like a radius. But keeping in mind that the deer location on these patches is not how it occurs in the real world.
Right.
Right. Yeah.
I mean, from a real world management, you know, like if I'm in charge of the targeted call, the center of the square is where I found that one. But then your numbers that you had, I mean, what was the, you know, it was like 3,000 or something that you'd have to call, you know, like plus minus whatever your standard deviation was.
Implemented over a period of 10 years. So again,
so that's over 10 years. That's 10 not in 1 year.
Not 1 year. No.
Okay, I was like, are there even that many deer there to shoot?
That was over 10 years. And again, I guess, Tiffany, you are exactly right. This was done just to get them started on this scenarios. The expectation was or is that they come up exactly with the way you said. This is not practical. This doesn't happen in the real world. We need a lot more practical kind of nuanced scenario where, you know, 1 year we are able to do the cull, the next year we have less response, and we can incorporate all of that, right? Unless and until these people are involved in the scenario design, it will end up into being something that is not connected to the real world. So that is, again, just kind of emphasizing the need for them to be driving this kind of,
more questions.
Yeah.
On your where you had low, medium, or high culling density, like what were those numbers? So roughly based on?
Low is 2 per square mile.
Okay.
Medium is 5 per square mile, and high is 10 per square mile.
Was there like a DNR or management group that you said you were able to say like, this group was really aggressive, they did 10. That's how I'm setting high?
Again, I guess this was in a very early phase in discussion with the agency, and and we were able to come to this kind of agreement after about 6 months of discussion, right? Because translating what happens in the model to a deer manager is not straightforward. Like you are exactly right. They come up exactly with the same question. No, but we don't. We are not able to do all of the deer. If there is a landowner which does not we have access, then we don't do that. My answer to that is that stochasticity is incorporated in here because you don't say which one is detected. Every time step, a deer has some probability of having CWD, another probability of being harvested, and then a probability of being tested. Right? So we have a lot of, all of that is if you do a high number of iterations, you kind of get all of that uncertainty in these scenarios. But I completely agree, it can be refined. And the agency people in this case, you know, we came to the 2, 5, 10 after 3 rounds of different scenarios. So it was not 2, 5, 10 earlier on. And I think how we did it was we looked at their actual data of actual number of deer with targeted culling data was available for 10 years. And we tried to get that into the model.
what if you went a lot higher? Like, I feel like 2.5 deer is your targeted call.
Okay.
I mean, like, I might shoot 2.5 deer on my own as a single hunter.
Right.
I mean, we're talking 640
Yep.
acres.
Yeah.
I go gun season and bow season. That seems like an incredibly low.
Okay. Okay.
You know, you aren't even going out for.
Right. Yeah.
It seems to me like that's not.
I'm looking in your paper right now, trying to remember how you set up. Are your deer moving between patches?
Okay. So the only deer movement we have in the model is when they disperse.
Okay.
Okay. Otherwise, we do have some deer, we have group dynamics in there. So sometimes the group kind of disbands, right? And some deer become solitary, some solitary deer join the groups. Like, so there is that dynamics. So whenever there is group dynamics, they might change their location. Otherwise, what we have—
Is that seasonal movement or dispersal?
No, dispersal is just like for males, it is twice
Yeah.
a year. For females, it is once a year.
Okay.
But
so this is seasonal.
This can happen when the group dynamics changes, and that is seasonal.
Okay.
Like especially after the reproduction kind of happens, or just before rutting and all of that. The main thing is what we have is like the neighboring groups interacting with each other, right? So the probability of neighboring those social groups interacting with each other changes seasonally. And recently we are analyzing a scenario, like, scenarios, we are evaluating public practices for their impact on the establishment and spread of CWD, like baiting, supplemental feeding, and carcass movement, right? So how risky are these practices? They're banned, right? When you detect CWD, agency usually goes out and says no baiting, or no supplemental feeding, or no feeders, and all of that. One thing is always of compliance. Like, we don't know if it's 100% compliance, one. But the second part is, can we use this to evaluate the relative impact of these practices? So for just to give you a movement example, it is very difficult to come up with a scenario unless and until you exactly know how many feeders are there in this count, right? But one way, a roundabout way to that, and again, because we are not predicting anything, we want to compare between two different scenarios, right? So a couple of papers talk about not all deer visit feeders. They came up with a value for their area of 1% of the deer visit feeders. So let's just make an assumption. You have sufficient feeders where 1% of the deer population is visiting these year-round feeders, right? And now what we are just looking at, if we change the frequency of neighboring those social groups contacting each other with these feeders available, so 1% of the deer going to the feeders, and these 1% deer result into extra connectivity between, or extra contacts between those social groups, or male-female contacts, which happen only during a particular season in the year. Now, because of the feeder, you have them like eating from one feeder several times in a month. Then there is a paper which talks about dominant males displacing others from the feeder, but we have the contamination, right, with the prions in saliva and all of that. So we can kind of come to a basic understanding to set up these scenarios. And we did that, and we did find some very interesting kind of non-intuitive outcomes. If you really assume a large-scale change in contact, yes, the CWD prevalence goes up. But as you fine-tune it to more realistic kind of settings, it becomes debatable whether, there of course must be a threshold beyond which you would have more transmission. But baiting, how many deer does it actually bring in to change the contact structure? I can do anything in the model, but unless and until we have some real world kind of data, some real world opinion or real world observation, which says that this is what baiting does. This is how, or maybe some camera tripping data. I think that is one thing we are really missing on, because a lot of camera trapping data has come out in the recent times, right? People have backyard, like they have the camera traps, and groups, they have a lot of data. can we use that to make up these scenarios is one of the things, I guess I'm curious about. Because anything to do with the modeling is actually goes back to what questions we are asking of the model and how we are setting it up. So, yeah. I just don't know. I know there are some other things, but I think we are nearly there. Just wanted to, the force of infection. I briefly discussed that. I am more, trying to convince the collaborators that we should look at the force of infection. And just one plot, I think, this makes it much easier to understand is you can also plot the populations, right? So this is again a 25-year projection for the same Linn-Macon County. And you notice that the number of CWD infected deer actually starts going down. That is not because of anything anybody did. It's just because the population started going down there. And so that came down. And so I usually present this data with a population plot, right? Should be somewhere here, but if not, then this is other, comparing different strategies. This is the 10-year data. So that number was for 10 years. So 10 years of post-season targeted culling, and then different scenarios, and then we can compare, which intervention made a lot of difference there. Finally, I guess, that's most of it. Yeah.
So
we have the surveillance model, but we can talk about it later. Yeah.
I was recently at a conference. I talked to someone from the Ohio DNR, and he was totally burned out.
Right.
He was like, this is insurmountable.
Yeah.
It does not matter, based on looking at similar models, what we do here, it's inevitable. We're just gonna have to live with this. So it's not the first time that I've heard about burnout on the agency side of things.
Right.
How can we best help? How can we best where's the optimism?
Right.
Where, how can we leverage all of this to really help advise the best approaches in the face of something that looks impossible?
And of course, I do not have the answer, or whatever, but I think more people coming together and talking about all of this and this cross-fertilization, but a one-sentence answer to this question, I think it's very important to address that part. It's very important. I think the answer lies in being locally relevant, but geographically coordinated. Meaning, if an agency just wants to do what 10 other states have done in the past, there is no evidence that that has worked. Yet they feel secure in having their response plan as a copy-paste from 4 other agencies. You open, I'm not making this up, open any response plan. It is based on what states before them had put out there. They might change some words, right? There is no evidence that has worked, yet they find it safer to do exactly what is in the response plans. I think what is really needed is we have to kind of adaptively respond to it. But the answer is in having locally relevant strategies. A strategy that worked for some state may not be of any use to Ohio. So that is where the phase-specific strategies, it becomes important. That is where having the characteristic of the local population incorporated into the decision-making process, that is important. And I think your work with all the state-of-art technologies, for them to implement those technologies will also require, I don't say my models, but any modeling approach will help us effectively implement that in a way where we can actually monitor the success. So far, there is a complete lack of monitoring how successful any strategy has been. It's all hearsay. Like New York is the best example. I have heard it for the last 10 years, but I haven't seen anything, a single letter published about it, a single paper about what is that strategy that worked, right? And so it's not blaming them. I'm just saying that, if it has worked, we need to kind of discuss that. And if it hasn't worked, we should not bring that example in. We should actually put out new stuff there. And new stuff is the only way we can, bring in optimism. Otherwise, it is like, I worry every day. Um, deer population management is entirely based on your harvest model. Hunter participation has been going down for at least a decade or more. Any new thing with CWD, the zoonotic thing, or even a false alarm, if that changes that hunter participation, then most regions will have a problem in controlling the deer population. Would the motor vehicle accidents go up? Would there be a lot more complaints and all of that? And then, I don't know, I am not so much, familiar with the hunting and all of that. But I do see, I don't want us to be in that place. And, preemptive approach. Yeah. Mm-hmm. No, I haven't.
Yeah, there's some information across different states.
Right. And, I went in and looked at their data. And I would say that you can check that too, but look at their surveillance numbers, abysmally low. And with such low surveillance, you are bound to miss detection, which can be falsely interpreted as the disease has disappeared. Right. Uh-huh. Yeah, yeah. Yeah. Right, right. Yeah. Yeah. Different. Exactly. Yeah. But I think like, even these open discussions, will help us go a long way with all of this, because think of the Western US. Like, nobody is talking about like multiple species now, kind of, we are still in a very beginning phase where it has reached Idaho, at least detected in Idaho. And, you have more species there interacting more frequently, west of Texas and all of that. So, do we always reinvent the wheel, or do we build up from what we have learned? This is again my very probably biased thing, but the European Union came up with that report, and that is a fantastic kind of summary and review of what has been done in North America. So I have to refer to their paper to look at what worked and what probably did not work and something like that. We could very well do that here as well. A periodic review of what's changing, what's happening, and particularly important with your kind of work. Because, I am wondering, like, why are people not queuing up in front of your office to take your technologies and do so many things,
I think the data that's been coming out of Mississippi and how, some of these technologies are being used in the environmental side is it's getting a lot of visibility. And I think people are seeing now that they can actually leverage some of these new techniques to, conduct surveillance efforts in different ways.
Yep. I do think these 1,600 counties, you are now providing them another tool to actually quantify, to actually be more confident about the detection of disease added to the hunter harvest. So what kind of bandwidth is available to do this on a very large scale, but a very programmatic, systematic way? I think that is one path that, again, it's way beyond my pay grade to even think of that. But I'm sure there are other people who are more kind of experienced into thinking about the big picture, right? But technology is there. But if you are not able to implement it, or if you're not willing to implement it and wait another 10 years before someone tries it and says, oh, this is good, then you lose the advantage. It is the only point I'm trying to make. Like,
any other questions from the audience? Zoom? Okay.
Thank you so much.
Fantastic. Thank you so much, Ani. I learned a lot, and I'm just, I get so excited every time I see Ani's presentations and the model that he works with, like this agent-based model, because I really think there's some great applications for it. So, Ani, thank you so much for the workshop today.
Thank you. Thank you. Thank you.
All right. With that, we're going to go ahead and log off. If there's any questions, please feel free to email Ani. I think we've